The next speaker is going to tell us a little bit about the AquAdvantage Salmon, food for thought. I have a feeling that Ron Stotish may never want to talk about this topic again. He must have been talking about it for quite some time now. And I'll bring him up. He is the head of AquaBounty Technologies, which he joined in 2006 as Vice President for Regulatory Affairs. He's also on the Board of Directors of BIO. He serves on the Food and Agricultural section there, and he's a member of many aquacultural associations and groups, so he's been thinking hard about this case study. So there he comes. So Ron, the floor is yours.
Thank you, Art. It's a pleasure to be here. I thank the organizers for inviting me. I will say, like Allison, when they contacted me, my first reaction was, oh no, not again. My second reaction was, as I understand the title and the scope of the meeting, we're not a laboratory animal. We're not a gene-edited animal. And I've been talking about this for the past more than 20 years, long before I joined AquaBounty. But I did realize that I feel a certain responsibility, a professional responsibility, because as a case study, we probably represent a history of this dialogue going back many years, as Allison pointed out. And I think that has value. And I think the discussion of the science that we do and the times that we live in. And I think it's important to point out that our views are driven by complex social and cultural issues, as well as personal values. And there are groups, individuals, and people who oppose every single issue that we deal with, whether they be issues of government, immigration, or science. And science is a particular challenge because most of our population doesn't understand where their food comes from, doesn't understand the technology that we use, and frankly is frightened by the prospects of some of the enormous technical capabilities that we have. On the other hand, science represents, and particularly the science of molecular genetics, an emerging science that has the opportunity to basically improve the quality of life for everyone, and it's doing so at a dramatic pace. The pace of research progress in this field in particular has, if you think about, we awarded a Nobel Prize in the 50s for the discovery of the structure of DNA, and nearly 75 years later, we're talking about genomic edits on a wide variety, a complete variety of organisms. So what I'm going to do today, and what I decided to do after talking with Allison and Alan, is basically tell you about our fish. And I realize that there'll be some things that we want to talk about, but I think you've all heard about the process that was used. I think you've heard about the approval process and so forth. But I really just like to take a little time and talk about our fish. I've been cautioned not to use this slide because it projects the impression that somehow we make gigantic salmon, and we don't. The reason we use this slide, and it happens to be one of the better pictures we have, is that these two fish are genetically identical, happen to come from the same spawn, are the same age, and they're sisters. The fish in the foreground is about 300 grams. The fish in the background is about 1 kilo. The only difference is in a single genetic locus. One fish carries a single copy of the AquAdvantage gene construct. That construct allows the fish to grow basically at a uniform rate from the time it hatches. The unmodified fish, basically, the growth hormone is under the control of a promoter, which is regulated by the photoperiod, water temperature, and food availability. So that regulation basically slows the growth of that fish, particularly in the early stages of life. Let's diverge briefly to talk about how these fish are raised commercially. And the first thing I want to say is if you eat an Atlantic salmon, and I had one for dinner last night, it's essentially a farmed animal. There are no commercial wild-caught fisheries of Atlantic salmon. There are Pacific wild salmon, and many of those alleged to be wild-caught. Some of them, about 50% of them, are what are called ranched, but Atlantic salmon are almost exclusively a farmed species. And predominantly, the farm technology is based on sea cages or net pens on the left side of the slide. That means they put these large installations in coastal waters. The fish are put in as young fish or smolts. They're fed, maintained by tender boats and by people who manage these farms. And then when they reach market weight, they're taken up by wellboats. They're either slaughtered on the boats and processed, or taken into factories close to the shore where they're processed as food. There are issues associated with salmon aquaculture as practiced in this way, but it does produce most of the world's high quality, nutritious, healthy Atlantic salmon. On the right-hand side is a facility actually that was designed as a smolt facility to grow young fish, a hatchery and smolt. And this is a land-based recirculating aquaculture system. Land-based systems are great because you don't have interaction with wild populations, you don't have interactions with coastal waterways, you don't have environmental issues associated with water pollution and other aspects. But the growth rates of the salmon and the unmodified salmon basically make these systems not economically viable. It's a nice way of saying that if you have slow growth rates and high costs of production, it doesn't make any sense to grow these fish in a land-based facility. The one thing that the AquAdvantage characteristic does is it changes that paradigm because it allows the fish to grow more rapidly. Let me remind you, we're talking about genome editing, and this is the sort of the Model T of this technology. And I say that not ashamedly, but this, as Allison pointed out, this fish was created in 1989. This is conventional transgenesis. The construct is a simple construct using a promoter from a cold water fish. And it's frequently referred to as a neolite creature, as though that were some sin in itself. But it's a promoter that the people who made this construct had and who knew the properties of. And they basically hooked it up to a structural gene for the growth hormone from Chinook salmon. That Chinook salmon growth hormone gene effectively produces the same protein that the Atlantic salmon produces. And the only difference about this gene construct is it's regulated differently than the endogenous promoter. And I'm not going to talk a lot about the science. I'm really going to talk about the fish today. That construct was micro-injected into salmon eggs. And for those of you who've done micro-injections, salmon eggs are like micro-injecting basketballs. So it's a lot easier than holding them with the little pipettes and using the Naroshige micromanipulators. Micro-injecting salmon eggs is almost a macro-injection. And basically in 1989, the people who did this work at Memorial University, Choi Yu and Garth Fletcher, selected a transgenic founder. They selected among the fish that took up the gene, the fish that were capable of passing it on to their progeny. It had entered the germline. And they also selected the fish that expressed a phenotype, the rapid growth rate. It was that fish that they began to breed, and it has been bred now for more than 12 generations in our hatcheries. It is not an overstatement to say that this is a food animal for which we have now a 25-year or 12-generation pedigree, and has probably been studied more than any Atlantic salmon, or for that matter, any food animal anywhere on the planet. We maintain this, and I'll show you how we maintain it a little later in my talk, but we maintain it in basically the male line, and we produce basically a population of fish that carry one copy of the gene. That's the commercial product. The phenotype of the animal, or in the case of the approved new animal drug application, the claim is that particularly in the early stages of growth, the animal grows faster than the non-modified comparator. As you can see here, the AquAdvantage reaches 500 grams in something around 200 days versus something approaching 400 days for the unmodified comparator. If you look at the growth rates later on, as the animals get larger and the growth rates slow, the growth rates actually begin to intersect. You don't get a bigger animal, it just reaches a target weight in a shorter time, and that's particularly true in the early phases of growth. We all know the ziggurat. There was a lovely slide yesterday that Malini showed that's much nicer than mine. This was copied from some of the FDA presentations, but sort of the escalating risk assessment paradigm that the FDA developed and published as Guidance 187 in 2009 was the eventual regulatory paradigm that we went through. And implicit in this is, after characterization of the construct and the sequences, a characterization of the phenotype, and in particular the durability. And because this was a talk about ethics of modified animals, I thought it important to remind you of some of the kinds of studies that were used to evaluate this animal. To begin with, in the durability studies, I have 3 or 4 slides here, and I'm not going to talk to the slides. I'm basically going to say, as I go through them, we measured everything that we could possibly think of measuring in a salmon. And you can see there, for instance, location in the water column, behavior, level of activity, predator avoidance. These were studies that the study directors were members of the faculty of UPEI and the UPEI Vet College. They were brought in as principal investigators on the study. In the old days, this would be called a target animal safety study. We don't use that terminology in this context anymore, but we basically looked at everything in a salmon that you could measure. We had a pathologist from UPEI take histopath samples that were collected, and basically they looked at every tissue. They looked at every tissue for physiological anomalies. They looked for abnormalities, differences in appearance, and any evidence of pathology. We measured histochemical or clinical analytes of every potential nature, and one of the interesting aspects of this is if you go to the physician and have a blood chemistry drawn, there are well-known norms for what your serum potassium might need to be, or your calcium, or your bilirubin. Those don't exist for most animals, and they certainly didn't exist for salmon. There were values in the literature, but we basically had to do comparators and to basically look at all the analytes and really look for outliers and look for extreme levels. And as I said, we basically measured everything that we could measure in a salmon. The conclusions are shown here, and I won't go through the whole thing except that, and the highlight is mine. There is sufficient information to support the safety of the construct in the AquAdvantage salmon. The proof of the pudding here is when you come to our hatchery and look in the tanks and see the fish. That erases all doubt that there is any negative impact on the fish. And if you see the non-modified comparators and the AquAdvantage salmon in the same context from the same spawn or same year class, the benefit of the AquAdvantage gene construct is readily apparent. There were some differences in the fish. Our fish have higher metabolic demands. They consume more oxygen. They're growing faster. They're less prone to avoid predators. They're really more interested in eating. And to a large extent, they're highly domesticated. They are not very reproductively successful in studies done by Moreau and Fleming and mirroring the studies of Devlin et al. At DFO. When fertile animals are exposed in artificial ecosystems, the growth hormone supplemented animals don't do as well as their non-modified comparators. They don't because they don't avoid predators. When food is limiting, there's no benefit to having an additional copy of the growth hormone gene because you need the food and the water temperature in order to be able to grow. So the benefit of the transgene is really only obvious in a cultured environment where you're able to supply the necessary temperature and nutrient levels. So in the wild, most likely, that would not be a benefit. Despite that, we were aware that people would be very concerned about the environmental aspects of this animal and potential environmental risk. So some of the things that we did, and I'm sorry, but before I do that, the other thing that we looked at was the composition. And again, we measured everything that we could measure. This is all available in the VMAC briefing packet from 2010. This was presented in a meeting in, I guess, September, October of 2010. And these are taken right out of the briefing packet. Basically, what the FDA concluded was it's an Atlantic salmon, and it's as safe to eat as any other Atlantic salmon. That finding has been contested publicly, and we can talk about that, I'm sure. But we've had 25 years experience with this fish. It's been in test kitchens, and it's been tasted by a significant number of people who are knowledgeable in the fish industry and knowledgeable about fish. And all I can tell you is this is a very nice Atlantic salmon. What safeguards did we build into the animal? You can do things in fish that you can't do in other species. We can do something called a homozygous neomale. That's sort of scientific double talk for a girl that produces sperm. The fish is genetically female, but it can be induced to produce sperm. Carrying the gene in that form allows you, when you do the mating, normal Mendelian genetics applying, as Allison pointed out, you get one copy of each AquAdvantage gene construct in each progeny then. We then took those eggs, and you can do something else in fish that is quite common and used in many commercial species. You can render those eggs triploid simply by exposing them to hydrostatic pressure. We validated the triploidy process to a high degree. Our batches generally run greater than 99%. The release limit that we were given in our FDA application was 95%. I think it's important to consider that the fact that these fish are all female genetically, and you can't fool Mendel and you can't fool the genetics, you can't change that, and the fact that they're to a large degree documented sterile, and that's documented by a fluorescence activated cell sorter assay on every batch, not unlike a release assay for a pharmaceutical product. And these fish are to be raised in land-based contained aquaculture systems. So you have 3 levels of redundant containment provisions that provide for an all-female population that can't reproduce with each other, can't reproduce with wild type, and are raised basically in tanks. So each of these steps is validated. Each of these was part of the application. And we hoped that by taking these extra precautions, even though there were environmental studies that said that the gene would probably likely die out in nature, even if fertile fish were deliberately introduced, we hoped that people would understand that because of the rapid growth rate, this fish enabled land-based systems. That fact, coupled with the fact that there were additional redundant biological control mechanisms, that this would render this an ideal fish to meet many of the needs of the existing salmon industry and address many of the concerns of the environmental community. Some of the other attributes, and this is from a paper by Tibbetts et al. In 2012. This was a nutrition study between 0 and 300 grams comparing AquAdvantage to the unmodified cohort. And in the top right of that graph is the thermal growth coefficient, and you can see that the AquAdvantage salmon grow significantly faster than the unmodified fish. Those are corrected for temperature, so it's a thermal growth coefficient. These fish really do grow faster, and that should be obvious to everyone, although that's contested, and it will probably be contested this morning. The feed conversion ratio is another important measure. In GH supplemented animals, it's typical to see an improved feed conversion ratio. AquAdvantage salmon has about a 20 to 25% better feed conversion ratio than the unmodified salmon. That means to get to the same target weight, you use 25% less feed. Importantly, and surprisingly, when we looked at nitrogen retention, protein nitrogen is one of the most expensive components in the feed and generally comes from things like fish oil and fish meal. We had a 5% better nitrogen retention in this study by Tibbetts et al. At the NRC in Halifax. And what that means is that not only are you 25% more efficient in feed conversions, but you're retaining better nitrogen. When you do the so-called fish in, fish out ratios, AquAdvantage has the potential to really change that dynamic and move way past the 2020 targets for the industry, becoming basically an environmental asset and perhaps the standard for the industry. So coming back to these systems, you have on the one hand the contemporary culture system, which produces a healthy fish and is a large industry. The global Atlantic salmon industry is a $10 billion industry. The U.S. alone imports over 300,000 tons of Atlantic salmon every year. On the right hand, you have a developing recirculating aquaculture system, which is great for raising young fish, but the economics are a bit dicey, to use a phrase that might be familiar to Allison, for instance. But let's look at the relative merits. And I'm not criticizing the sea cage industry. It is the established industry. But one of the reasons that AquAdvantage was developed is we thought there was an alternative. In the sea cage industry, you have exposure to pathogens, particularly bacterial diseases, viral diseases, and sea lice, which aren't really lice at all, but small crustaceans which attach themselves to fish. You also have the escape of fish. Sharks and seals get into these things, they rip the nets open, they eat the fish, they release the fish, and no one knows what the impact is when you release large numbers of fish. And every day you can pick up the trade papers and see where they've lost 50 or 100,000 fish in some part of the world. The use of medicated articles associated with those bacterial diseases right now has devastated the the salmon growing industry because of rickettsial disease in the Chilean industry. They're just recovering from an infectious salmon anemia outbreak. And the point of that is that the fish that are in those net cages, and again, proper management and biosecurity can make that system work well, and it does produce a healthy product, but it does expose the fish in both directions. The caged fish are exposed to the pathogens from the wild fish, and the wild fish, even from different species, are exposed to the pathogens that may be present in the caged species. So, and last but not least, as someone standing here with a bad cold this morning that I got from my granddaughter who was in daycare, I can tell you there's no such thing as one sick organism when you have lots of animals in one place. And it doesn't matter whether it's a fish, a human, a dog, a cat, or whatever. And that's something that also gets lost in a lot of these dialogues. So the elements of having a controlled environment and recirculating aquaculture doesn't preclude an infectious disease, but it certainly presents an opportunity to mitigate it, prevent the spread, to treat it, and to recover. In the operation and transportation, and I'll talk about that briefly, these fish are now grown in areas of the world such as the south of Chile and the north of Norway, Faroe Islands, Scotland, and so forth. Those fish are then, the fresh fillets, are put into 747s and flown to New York or Miami. The carbon footprint associated with that is enormous. If you could build recirculating aquaculture facilities, and let's just arbitrarily say within 500 miles of every large American city, you could slash that carbon footprint significantly. We weren't quite sure how to do that, but we went to an ag economist in London and said, here are the two alternatives, and understand that we're presently approved only for Panama and for PEI. So this is a hypothetical slide. In the event that we were able to put a large facility to produce salmon within 500 miles of an American city, we could reduce the carbon footprint of transportation, and this is only of transportation, by more than 20-fold, perhaps 25-fold. That's a significant savings. It also would reduce the cost of transportation by more than $1, $1.50 per kilo. If you think about 300,000 tons times $1, $1.50 per kilo, that's an enormous savings in time and carbon. So, the land-based systems have a disadvantage in capital cost, a large one. It's roughly $10 per kilo, 1,000-ton facility a year, you're talking $10 million to construct. However, if you can work in an area where you have a low energy cost and the cost of running the pumps and the rest of the operation, and if you have a fish that grows in a desirable rate or at a desirable rate, you have the potential, for instance, of improved feed conversions. You've reduced the need or the possibility for exposure to other animals. You've reduced the need for medication. You've reduced the transportation cost, effectively zero impact on biodiversity. And last but not least, you could have fish that were, instead of being 10 days old by the time they showed up in your store or your retailer, they could be 2 or 3 days old, and at a lot lower cost, both in terms of carbon and in terms of real dollars and to get them to the market. So we think that the challenges, and there's no one solution for global food production. The needs for aquaculture are significant because aquaculture can present a safe, high-protein diet at a very efficient level of production to the emerging global population. The needs of the developing world, and if you talk to Africans, they think Americans are quite silly. Aquaculture for them is food and survival. Aquaculture for us is something that other people do. Less than 5% of the seafood we consume in the United States is produced here. 95% is imported, and it's imported sometimes from places that we probably would not like to think about, and using technologies that we probably would not like to hear about. So we have an opportunity. To use this type of approach, which I think gene editing will only expand and improve the opportunities to address a lot of the needs, both environmental and economic, to meet the needs of a growing and developing world. With that, I'll stop, and I think we'll go on to the next talk. And if Art will allow me, this is misbranding, and I'm sure that Laura and Larissa will say so. Our approved product is an egg, but I put the NADA number on the salmon because I think it's important that we remember we're talking about food here. Thank you.